Go. I think this is a very nice countdown. Alrighty. Welcome in to this fine morning. It's actually beautiful here in Houston. I believe it's also nice over there in over at Stanford. Today, I'm joined by Professor Noah Burns of Stanford University. He was going to, come on, speak to a little bit of his chemistry today. It's really rock solid. I think one of the benefits to your chemistry is that, while also being rock solid, it's like you're able to make these very mouth-watering, eye candy-looking structures that looks good to like the chemist. So like those laterines, like it's just like, as someone who's like not a total synthesis guy and like chemist, it just like looks really cool, you know?
Yeah.
And today we'll be talking natural product stuff. I guess before we go there though,
Sure.
I kind of want to talk to you because you're from Maine. You're born in California, but grew up in Maine. So I'd like to talk to you about that because Maine is probably like, I don't know, top 3 states I want to go visit, like there and like Colorado and like Montana.
Right on.
So, you know, how was growing up in Maine? Are you from the Portland area or more like the backwoods?
I'm from,
Maine?
It's close to Portland. It's about 30 minutes inland. So it's not right on the coast, but, close enough. I mean, we spent a lot of time in Portland. But
it's like the only city that anyone knows in Portland.
Yeah, no, totally. And I mean, it's an amazing city. It really is. And I kind of consider myself like not from like deep Maine, is like further up once you go up to Bangor, and then there's like a huge amount of state above that. But, most people live in the sort of bottom triangle or so. And, it's not super remote, but it's Maine. I mean, got to drive everywhere. The weather can be great or absolutely terrible. You got to be a little more creative about things that you do. You know what I mean? I mean, I grew up exploring the woods and, we moved to be on a lake when I was in middle school, and that was amazing.
I just feel like the landscape is just breathtaking up there. Like during the fall, like I assume like all the the colors, the trees are changing.
Yeah.
Okay. I don't know. I don't know if this is it's not really a hot take, but this is a little prejudice here. But how like important is the like the lobster business and like crabbing and stuff like that? Is that like a real thing up there?
I mean, the lobster business for sure. There's a community of lobster fishermen, all along the coast that are, they're kind of beholden to, regulations for how much they can fish each year, and that results in the price of things. It's not one of these fishing markets. I mean, I can't say that I've followed it super well in the last few years, but, it was pretty restricted for a while because of, worries about the population, but it kind of bounced back. And so I don't know what lobster prices are at right now, but you can kind of look at, how the fishermen are doing based on that.
Something that still doesn't necessarily make sense to me is like why lobsters cost so much. So it's like, they're literally like the sea bug of like the ocean. But like
prisoners used to eat them, right? That's like the joke.
So, Maine, just a beautiful city or beautiful state when I get up there. What's the cultural like up there? Like what sort of influences are there? Like, let's say in like the, over in like Portland and like, the greater Maine area, like what kind of like cultures you kind of see up there?
There's a little bit of sort of Canadian-ishness to it, I would say. I mean, my sister actually moved to, she went to McGill and she's stayed in Canada. So I've spent a lot of time in Canada now, and that's made me kind of recognize it. I mean, and I mean that in like, people are really nice. I mean, it's obviously like a little slower, way of living than like in the Bay Area. But the people are really wonderful. And Portland's turned into this amazing like food city destination, which is really like there's a lot of culture there. If you like that kind of city aspect. I mean, I grew up in a community of like 50,000 people that was maybe a little bit on the conservative side, and I don't know. I mean, we had to be a little more creative about how to fill one's time. There's not all this sort of sensory overload like there, is in the Bay Area. I'm sure Houston is like that. I mean, You know what I mean?
Yeah, we do have the added benefit though of like, the energy capital of the nation and all the polymers and plastics you can possibly imagine. And so I guess from that sense, I guess it kind of transitions us nicely into like how you originally got into chemistry, because I can't I don't know, you said how do you fill one's time? I guess for you it was partly STEM, but I don't know.
Yeah.
So like middle school, high school, like how'd you get into it? And
it's a great question. I will say I didn't know I was going to be a chemist until sophomore year in college. I didn't, but science and math were always fun and fascinating to me.
Sure.
I think I was always a very curious and continue to be a very curious person, asking all the, silly questions when you're a little kid of why the sky's blue and all that. I just remember asking so many questions, and, starting to learn in science what the basis is for all these things. My son is 11, and, I'm looking at some of the stuff that he's doing, like in his science class, and they're looking at, they're doing a lot of earth science. And I remember loving that, learning about how the weather forms and, different levels of the atmosphere, which is really cool. But I don't know if this is the reason, but, I could say like the lack of all the, busyness of life.
Sure.
It kind of, results in me having to be a little more creative about how to fill my time. I talked about I mentioned kind of exploring the woods and stuff like that, but
yeah,
I spent a lot of time, and it's funny because people use this analogy for total synthesis all the time, but
yeah,
I spent a lot of time with Legos. I love Legos. I love, playing out, I don't know, science fiction kind of stories and things like that, I
love Legos, dude.
Science fiction lover, like that's always been something.
I honestly can't think of one STEM person like doesn't like Legos. Like honestly, like I will be,
right.
Like I like that, yeah. No, that's awesome.
But obviously, like you, like all the people we're around, like we had an aptitude for science and math, right? My father is he's a family doctor. My mom was a teacher. She's retired now, a high school and elementary school teacher. And, I thought maybe I'd be a doctor. It's
right.
It's a classic kind of boring story. So I took all these pre-med classes and, it was sophomore year when I took organic chemistry that I just really fell in love with it.
Yeah, fell in love with it. I was gonna ask you because, because you did your undergraduate at Columbia, did a little bit of undergraduate research, but how was that transition though? Let's say from Maine to like New York City. That's got to be
it was
an
crazy
experience.
No, it was really crazy. The good thing about Columbia, like I was curious about that change, right? And yeah, sure, maybe I went to the extreme, but like NYU's like in the city, like there isn't really a campus, and Columbia is cool because it is a campus. Everyone lives in housing, which is mostly on campus or, a block or two off campus. And so, when you're on campus, it's like this, it's almost like this walled kind of fortress. I don't know if you've ever visited Columbia, but,
not specifically, but I've been in New York City.
Yeah.
I know the vibe.
So right on. So it feels like university when you're there with like a campus and things, there's quads and stuff, but yeah, it has this access to, one of the greatest cities in the world, right? And everything that offers. And yeah, I spent a lot of time off campus. I had a friend from high school who went to the new school downtown. And that's a cool place because it's, they have like a writing program, they have like a jazz program, they have art program. And so in my first year or two, I actually spent a lot of time down there with some really, total non-science people.
Right.
And that's actually, it wasn't really until like college and then grad school that I started, even more so grad school, that, I started having good friends that were in the sciences. Like I spent a lot of time, some of my best, most of my best friends from growing up and stuff are teachers and chefs and, just really, I like being around all sorts of different people.
Right. I mean, especially in New York City. I mean, it's literally like, between musicians, artists, top-notch science, like it's all there. It really is.
All there. All there. I was done at the end. I will say that after the 4 years, I was like, okay, this is enough.
Don't you think that's like where every university you go to? Or do you think that's like especially true for like the...
I think you're right. No, I absolutely think you're right. I do,
Well, do you think, or let me ask you this then. So like the New York City in general, like, I think New York City is great for, let's say, if you're kind of a young student to be there on your own and explore. I don't know if I'd necessarily raise a family there in New York City, but that goes for like all major cities though.
Yeah.
Like one day when I have a family, I don't see myself living in Houston. Like I don't see myself living in New York City. I'm from a suburb, so like kind of like you're from the suburbs area. So like that's where I envision myself.
Yeah.
So I think it's good to, but like New York City from like 18 to 22 sounds like a great time though. It does.
It was. It was. And, being able to live there, unless you start out right out of college, in one of these, I don't know, some top paying finance job or consulting job or something like, living in New York and not having very much money, once you're older than in college, that sounds kind of tough to me. You know what I mean? Like,
yeah, that's still the appeal of it. Right. So it's like, like kind of just making it out in New York City. That's some of the American dream right there. Just making it, passing by.
Yeah.
But you also did undergraduate research with Professor James Leighton, I believe, right?
Yeah.
So, I guess, you said you didn't really, like myself, I didn't really get into chemistry. Like we all took our gen chems, I guess, that freshman year, but organic chemistry is like where it really started to click.
Yeah.
And I really thought for me personally, this could be a real thing to pursue. I envisioned probably similar pathway for you if you're, saying organic chemistry sophomore year. So, what was that process like doing undergraduate research? Because it can be, I know a lot of undergraduates who are like afraid to kind of take that step to go ask to do research in like a research lab. And so, how was that transition like? And, what did you work on too?
Yeah. I will say that experience really defined my career,
Right.
I can't like over kind of emphasize it in a way. I took Gen Chem. I didn't really like Gen Chem. To be honest, it wasn't.
I don't really know if anyone actually does. Like that's like seriously, like
even the professors, like my colleagues, nobody wants to teach Gen Chem either.
Right.
But I did. I did very well in Gen Chem and established a relationship with a physical chemistry professor. And so I did some like work study for him, just working on his PowerPoint slides, and adding animations and things like that.
Gotta start somewhere, man.
And then he, I was like, I want to try research. And so, like a lot of places, they have an undergrad research program. So I worked for a summer at the end of my freshman year. I worked for a summer in his lab doing scanning tunneling microscopy, which basically involved like sitting in a 50-degree room for, 6 hours a day, looking at this, looking on the computer, trying to get good data. Physical chemistry, I didn't love it, I didn't, but I think it did have a good impact on me because it just showed me that, like you were talking about this, some people have this barrier of like, how do I get into this? Or like being, I don't know why, people are intimidated by professors or whatever that, but I saw that it was possible. Columbia is a place that doesn't, like Stanford actually, that, it doesn't have a ton of chemistry undergrads. So
right.
like the, ratio of faculty to majors is high. And yeah, I just saw that. And so it was like, great. I'm happy about this. And then it was, when I took organic chemistry and really started falling in love with it and kind of asked around, other undergrads that were doing research, like, what for advice on what lab to maybe join. And yeah, Jim Leighton taught my, the second semester organic. And I asked him and he said, yeah, let's do the summer program. And, I was definitely like deer in the headlights when I first got there. Like we all are, you know what I mean? Like, it was like,
you mean totally... you didn't know all the answers before going in the lab? Are you kidding me?
No,
how are you professor, man? You literally, you didn't know all the answers before going in the lab.
Absolutely not. I mean, I don't know. I didn't know my ass from my elbow, right? I mean, you're learning the techniques, but it's like,
dude, you got to learn the glassware. Like people, like it's a real thing. You have to learn how to use a rotovap. Like these things are real, man.
Yeah, yeah. And translating from your undergrad lab classes to like a research lab it's very different.
It's very different.
But very, very early on, I was like pretty captivated by it because, I love thinking about organic chemistry. I mean, obviously, the material, the topics, thinking about chemical reactions, thinking about, it was a lot of the spatial intelligence side of organic chemistry that really appealed to me, starting from the bonding, but then going into three-dimensional structures, talking about chirality, that, and yeah, whatever, that's a connection to to the Legos, right? Liking to build things in 3 dimensions. But then starting in a lab and then, you learn about what a Grignard reaction is, and then actually, doing that with your own hands in the lab, that was, that really captivated me. I'll say.
There definitely is something to be said about because I took organic chemistry that fall of like whenever my sophomore year was. And then I did, undergraduate research the following summer. Uh-huh. And so there's something about like seeing it on paper and it's like, and I guess early on, it's like, you're kind of just memorizing these reactions in some sense where it's like, you're just kind of like just trying to do the thing. But then it's like, oh, wow, I actually just made the Grignard and I actually just like, attacked the carbonyl or something like that. It's like, it's really, something special to be said about like those first, like,
right.
I don't know, putting your words into action, I guess,
Right. And, that's not true about all science, I think.
Right.
I love doing things with my hands. I really, that's one of the things I miss about being at this stage of my career is that, I do not get to do that.
You're writing now. You got to write and you write grants and stuff.
I'm always like, I'm going to, maybe this summer I'll get back into the lab or something, but
yeah,
I don't know. I'll probably just make a fool of myself.
Yeah. So you did research with Professor James Leighton for a while, but then you ended up going to the Scripps Research Institute, right, with Professor Phil Baran.
Yeah.
So, I think that's a huge jump personally.
Yeah.
So, can you take me through that? Because,
sure.
Honestly, like when I was applying to graduate school, like, I didn't know, you don't know who the big players are, I guess, in a I didn't know who the big players are in the chemistry field until like you actually get there and it's like, oh wow, here are the big names. Professor Phil Baran is a big name, especially in total synthesis stuff. And so, like, I guess I'm curious to know, did you know, he was one of the big players in total synthesis before that, or like how, like, and so like, how did you know you wanted to go to grad school? So all the above.
Yeah. I definitely want to say that, Jim Leighton had a huge impact on me as far as a mentor, how to think about science, but, really also as a person. I really looked up to him, and it was seeing him and working in his lab that it was then that I decided, like, I want to be a professor. Like, I basically wanted to be him and put my blinders on and just went for it. Okay.
That's really awesome. By the way, have you talked to him since being professorship? Has it come full circle?
It's been a little while right now. I haven't talked to him since before the pandemic.
Okay.
But he came out here for a talk a little while after I started here. I visited Columbia when I was on my sort of tenure tour. That was great to see him there.
Full circle, man. I love those. I love hearing those stories.
It's cool. No, it's cool. And I think pretty weird for him too. You know what I mean? Like, you're this little undergrad and now you're an assistant professor. Like,
That's really cool.
But, I initially worked on a methods project. It was a alylation of ketones. It was basically extending some of their strained silocycle alylation chemistry. So, I worked on methodology for a little bit. And then there were total synthesis projects going on in his lab. And, I asked him at one point, could I work on one of these? I mean, I was kind of intrigued by it. And so for the, last, I don't know, year or so in his lab, that's what I did. I mean, I worked on a total synthesis project. And that's when I really, again, fell in love with total synthesis.
Sure.
And really wanted to do that as a grad student. Now, Phil Baran was, when I was an undergrad, he was finishing up grad school essentially. And, I was lucky to get good advice from grad students and see that, just learn to, as an undergrad, to really try to follow the literature. And it was, because total synthesis, I was very curious about it. I just started really following a lot of that literature more so, when total synthesis papers would come out, I would read them. And there was this guy that was, I don't know, he published some crazy number of papers in his grad career,
sure.
Something insane. And so people knew that he was like a, sort of up and coming person, right? Now, when I applied to grad school and visited grad schools, he was in his first year. So, people knew, oh, this is this exciting guy, but who he's not the
who knows
giant that he is now.
Right.
And when I visited there, again, I want to do total synthesis. And when I visited there in this first year lab, I met this guy that was so excited and enthusiastic about the science and had attracted this group of, he had a group of maybe 6 or 8 grad students when I visited, and he had attracted all these people that were just as excited. And it was really contagious, and it was people like, Ryan Shenvey was there, who's at Scripps now, right? Tom Maimoni joined pretty soon after I was there. Tim Newhouse came later. Just a lot of amazing, Ian Seipel. It kind of snowballed. He and I learned from that too. He recruited just really great people, and they really. He set the tone, and the group. I don't want to say ran itself, but the group had its own kind of mentality that came out of that.
The way you're describing it just seems like everyone was just driven to do like total synthesis. Like it just something about, and by the way, maybe we should explain total synthesis a little bit.
Sure.
For people I'll let you kind of take the wheel on that.
Sure.
It's not really my area.
Right.
I'll let you take the wheel. And if you want to explain total synthesis, I'll let you take the go at
it. Sure. In its sort of, I don't know, basic form, it's trying to make small molecules that come from nature and make them from scratch in a lab with chemicals you can buy off the shelf, right? People go out and they isolate these small molecules from anywhere, from plants, from, there's a lot of marine natural products.
The algae on the seaboard.
Sea sponges and algae species and things. And, a lot of the purpose of that to, it's not just for fun, right? People go out and they essentially, extract out, whatever, they take an extract and maybe, try to treat different types of diseases with that, right? The classic is like you shoot it at some cancer cells and see if it kills them. That's the sort of most basic, or at bacteria and see if it kills them, right? And then when there's leads, people will actually try to figure out what are the components of that, right? What are the specific molecules that are leading to those maybe exciting effects or biological activity? Another reason people do this, and there's been more of this in recent years, is, based on traditional medicines, like traditional Chinese medicine, right? I mean,
Right.
people have been using plants and, natural extracts for, even thousands of years, and, without even knowing what's in them, but it's all based on anecdotal evidence, right? I mean, Chinese medicine is very different than, Western medicine, but there's a lot of, there's effectiveness there, and
trial and error.
Finding out what the actual molecule is that leads to that is really beneficial, right? So, I mean, people isolate these things, and then, most often they only get a little bit of them, right? And then it's like, okay, well, how are we going to keep researching these compounds? And that's where, right, total synthesis comes in. I've always thought it's kind of a funny term, total synthesis. I mean, you got chemical synthesis, I don't know, whatever.
It sounds extravagant, though. I'm not going to lie. Like, total synthesis, it just sounds extravagant.
Yeah, no, exactly. I remember telling one of my friends in college that, I'm doing total synthesis, and they were like, total synthesis. It's like, yeah, it sounds kind of funny.
Yeah, it does sound. And I will say, for anyone that's like, I don't know, this is actually, no, I'm gonna say it. Sure. If you want an intro to total synthesis, I mean, the book Classics in Total Synthesis has a lot of very pretty pictures. You don't have to understand the chemistry. That's fine. It has a lot of pretty pictures, and it explains it'll explain all the natural products, why they're important, and why one would want to make this.
Yes.
And that's a great introduction, I think, to total synthesis.
You know what's another great one? I don't know if you also by Casey Nicolau is this book called Molecules That Changed the World.
Okay, I've not heard of that one, but
you should check it. I mean, and for the
viewers too,
because it is written for like a general audience more. It doesn't talk about total synthesis as much. So classics is, I mean, it's
classics is definitely a chemistry's like handbook. But if you want 100%. a more general approach and a more non-chemistry friendly, recommend. What's it called again? Moleculars.
It's called Molecules That Changed the World. Yeah,
okay.
I have a copy.
I'll add that. I'll add a link to that in the description here.
Cool.
But here's the thing, though. During graduate school, it wasn't all hunky dory. There's no way, you were like getting good results the first few runs.
I'm not going to paint this rose-colored glasses.
Yeah,
it was all fun and games all the time.
Yeah. Despite contrary belief, many chemists don't get their natural product first attempt. There's a lot of trial and error, a lot of trial and error, but also a lot of critical thinking about why these things aren't working and why some transformations aren't working. Yeah. And so I'm curious to know, like, what was your mindset going into those sort of scenarios? And, because I'm sure, especially, in those first few attempts when things weren't working, you probably hadn't experienced anything like that before. So, like, whether it was like advice from other graduate students or Professor Phil Baran, like, what was kind of the mentality there?
Yeah. Yeah.
And, the second question to that is, when did you remember that first eureka moment where, like, you finally got something to click and, like, and then from there, everything seemed to start working a lot. So I'm kind of curious to see those two.
These are great questions, Aidan. These are really great questions and really fun to talk about. Yeah. So, I mean, that's that, and I like you touched on it. Like, that is the challenge of it is the fact that, everybody comes into grad school, having been really good and successful in their classes and things. Right. And, that type of success is, I don't want to say easy, but it's clear and the goal is clear and the pathway to get there is clear, right? You study hard, you learn the material, you're going to do well, right? But when you get to your PhD, that's usually for people, that's the first time where you have this problem that has never been solved before, and you need to figure out how to solve it. And that can be really terrifying, I mean, part of my job now is to try to kind of, is to advise people to do that, right? And, some people really struggle with that more than others. but that was a real eye opener, and it was scary. It was scary to me, because it was like, shit, like, I mean, sorry, how do I do this? You know, how do I solve this problem?
Yeah.
And you also kind of said it, you know, it's, you know, other students is a really, you know, that's you can't overemphasize that as a resource, right? You got to talk to people,
right.
And, you know, people that had some, that, you know, were having some success and had, you know, broken through and gotten there. Like, that was very, very, very helpful. But Phil was fantastic. You know, I mean, I was very lucky at the time to, you know, be in his lab early on when it was small, and he was there all the time. I mean, my hood was literally like right outside his office. And so I could just suck all this information from him, you know what I mean? And advice. I mean, and that is one of the reasons that I went there versus going to a more established professor, because I realized that I would get the most face time with them and that I would learn the most. I mean, that really was like, I mean, it was the environment, but it was this recognition that I'm going to get to spend a lot of time with this person, and just, you know, this is my opportunity from my viewpoint. When I looked at my, you know, my whatever opportunities, this was the one where I said I could learn the most. And so, you know, learning, you know, you have to learn so many little practical things. I mean, how to run a column, how to run a column quickly, you know, how to run a TLC, how to gain much information. You know, TLC seems simple, but you can gain so much information from a TLC, right?
Yes.
And just, you know, kind of going through that process of solving the little problems along the way.
Sometimes just changing a solvent, it just works. Like just something simple, and you don't have any reason why that would work. It just does. And and it's that, you know, I mean, this can be the difference, and you know, this may be, you know, the difference between, you know, success and not being successful is really keen attention to detail. Yeah.
You know, I mean, you got to notice the tiny little things. I mean, you said it, like, you know, changing a solvent, you know, just looking at what does reaction physically look like, right? Just, I mean, even just staring at it, right? And noticing a color change, and, you know, that can mean something, and color change is indicative of some chemical.
Right? It means something, right?
I mean, Phil used to say, you know, he has so many good quotes, but, you know, he used to say, like, you know, reaction needs care and love. Like, you got to treat it like, you know, it's not something you kind of slap together and leave. You got to really kind of give it some love, right? But he also, you know, just as, I mean, I was talking about these little kind of details, which are very, very important, but you have to keep your eye on, you know, you have to be paying attention to little details, but you got to be looking at the big picture, right? I mean, you know, what is the problem we're trying to solve? Well, in total synthesis, the goal is clear. You want to make this molecule, but, you know, it's an advanced science in a way, in that, you know, if a molecule exists, if it can physically exist, we know we can make it. You know, that wasn't clear in, you know, the mid 1900s. I mean, R.B. Woodward, right, was a real pioneer in a way, because as far as complex molecules are concerned, you know, he showed that they could be made. And all the giants that have come after that, right, have shown that anything can be made. But, you know, Phil really tried to push us in trying to make things in ways that are more efficient, more exciting, and you know, lead to new chemistry, right?
Yeah.
I mean, the way we put molecules together in total synthesis is we string together, you know, chemical reactions, and there's a lot of known chemical reactions. So, you know, you could just string together all these known chemical reactions and get there. And that can be fine sometimes. I mean, you know,
right.
I mean, that's what the pharmaceutical industry does. You know, they're not trying to invent some new crazy reactions to make a drug. They want to use things that are very well established. They know they're going to work, right? And I mean, all this, you know, synthesis planning that I mean, it's been going on for a while, but is really, on the ups now with all the, machine learning and AI, right? I mean, that's what they can do. They can tell you, or trying to do, right? Tell you how to make a molecule based on known science. But one of the really cool things and benefits of total synthesis is you're making a complex molecule. So you're doing reactions in kind of new settings. And that really inevitably, I mean, even if you push yourself or not, inevitably is going to, lead to advancements in chemical reactions.
Yeah.
And it's a little bit of a frustration to me. I don't know. I don't want to be griping too much here, but it's a little frustrating to me because the day of being able to, make a molecule just for the sake of doing it is, I mean, I'll put it this way. If you say that to a funding agency, they're not going to give you money.
Right.
Which is fine. I mean, I mean, again, I don't want to sort of argue, whatever.
Yeah. But so in graduate school, like what did you like? What types of natural products were you working on? And what were some of the difficulties that you had found personally? But then obviously you made it, so you did something right.
Yeah. But there was a long time where I didn't think I was going to. Right. I mean, to talk about the sort of personal side of it. Yeah. I mean, it wasn't all, all roses all the time. Yeah. Right? It's really hard, and having been successful for some years, you can start to doubt how good of a scientist you are, how good of a chemist you are, whether you chose the right pathway or not. Right. I mean, every grad student has these, I don't know, big questions go through their mind. I don't know anybody that at some point didn't think, maybe this isn't for me. Maybe I should try something else.
Yeah.
Because it's so hard.
I still say to myself, it's like, just because your chemistry doesn't work, it doesn't make you a bad person. It's like,
yes,
that's what it always boils down to.
Yes.
You are really on the cutting edge of science. It just is what it is.
Yes. Yes, exactly. I mean, nobody made an advance in any science, any field, without so much failure. I mean, I don't know what the statistic is, but 90% of reactions you run are going to fail. Right.
Yeah.
And I mean, that's really disheartening, but I will say this, and, I think this is a characteristic I've seen in a lot of successful chemists, organic chemists, is eternal optimism.
Yeah.
Yeah. I mean, again, everyone's going to get down at some point, but you got to think this next one is going to work. Right.
Yeah.
Oh, I mean, like you said, I use this solvent. Well, oh no, that solvent for these reasons that I think in my mind, that's going to do it. Yeah. And then you just go do it. And then, okay, it doesn't work. But then, this next change that I do is going to do it. And if you,
it's a good mindset.
If you do, it's a mindset. It is a mindset. It is a mindset.
So I'm going to ask you this. So when you weren't doing chemistry, what were some of the things you like to do out in, Scripps where is it? It's California,
it's in San Diego. It's
San Diego.
Okay. It's in La Jolla, which is just a small place. It's right near UCSD, but it's not. I mean, it's its own independent research institute.
Yeah. So outside of chemistry, what were some of the things to do out there, and some of your, and also, the comparison, because New York City to San Diego. I mean, how the East Coast versus West Coast, what some of those changes there as well.
It was a change. It was a change. And I mean, I'll be honest, I'm not somebody that I've made a lot of big changes in my life. We could just say geographically, but I'm not somebody that change is hard. It is hard. And it was another kind of culture shock, from New York. And it was a little bit initially there's not as much to do here. I don't know. I mean, I did, at least my first couple of years, for better or worse, I spent essentially, most of my time in lab. You know,
Sure.
I really did. And, you know, Phil never, I mean, nobody does this today, but, you know, there were times when people, you know, professors set hours and they want you there for this time. And Phil never did that. I've never done that in my lab. And that's one of the beauties of being a grad student, right?
Yeah. You can kind of carve out your own hours, but, you know, you say, obviously you gotta work hard, but, can't slack off. But I mean, you can basically carve your own hours, though, whatever kind of works for you, for sure.
Which is super cool. So I really came to appreciate the outdoors that it offered. I mean, you know, you can drive a few hours and be in the desert. You can drive, you know, less than an hour and be in Mexico.
Oh, wow. Okay.
Yeah.
Did you ever do that? Did you ever go to Mexico?
I did.
Oh, wow.
I did. I spent a decent amount of time down in Mexico. What's that?
I was gonna say when my mom used to be, she used to go to UNLV, and she used to go to like over to that western Mexico border. Is Tijuana over there? Is that the city that's over there?
That's it. It's right across the border from San Diego. I mean, literally, like, you know, they're almost kind of like sister cities in a way.
Yeah. My mom was telling me she had like the best like street tacos there, like ever in Tijuana.
The food.
So did you go to Tijuana though?
I went a little bit to Tijuana, but I tended to drive kind of south of there. I mean,
fair enough.
Rosarito is a neat place. Ensenada is like beautiful, beautiful, beautiful. And, you know, you drive down along the coast, which is beautiful. And I mean, the seafood is amazing. And, you know, it's inexpensive. So as a grad student, it was great. I did a lot of camping. You know, I did pick up surfing later on in my third year or something like that. And really fell in love with that.
Yeah. So. You still surf now? Or like, is it kind of hard? Okay.
I would love to. I'm always like, maybe I'll, because
get out there.
Pacifica here in the Bay Area, you could go, but it's a lot colder though. It's a lot colder.
Yeah.
The water here.
Yeah. But yeah, it's, I know the scenery out in especially like the South LA, or not LA, South SoCal is, I mean, just beautiful.
Yeah.
One of these days I'll get out there. But,
you should. No, it is really, you know, come out here and get a car and drive, you know, drive down the one from San Francisco to LA. That I recommend.
Yeah. We had one of our old graduate students who graduated not too recently. He grew up in Miami, but he loves going to like California. And he tells me all the time, like he drove from like San Francisco basically to LA, and like just driving that. I mean, I can't, he was like, I mean, just me thinking about it, like I can't think of a better drive. Like, you know, just the scenery. But, so, okay. Yeah. So you're at Scripps for a while, working professor. And then you also mentioned too, because of the influence of your undergraduate professor, you knew one day you wanted to become a professor. So,
that's right.
You know, obviously, well, maybe not obviously to some people, but you know, you got to do a postdoc, I guess. I guess that's just the natural order of things.
You do. Yeah.
But you did it with Professor Eric Jacobsen, I believe.
Yes.
Correct. So, Harvard?
That's right.
Harvard. Okay. I guess, you know, so what attracted you to go, you know, do a postdoc with Professor Jacobsen? And, you know, what did you work on as a postdoc, and how was that experience up there in Harvard?
You know, it does tie back into my undergrad advisor in some ways. I mean, Jim Leighton, in that, you know, Jim had worked with Eric Jacobsen as a postdoc. And I think, you know, he was definitely, I mean, very much influenced by that. So, you know, Eric Jacobsen's research program has always, it's not necessarily, I mean, they've done some total synthesis, but that's never been, I mean, anything of a focus there.
Right.
You know, their focus is on, I mean, you know, as I said, right, total synthesis is made up of this sequence of individual reactions. And his lab has always been focused on the development of the individual, you know, one specific reaction, finding a better way to do one transformation, you know, A to B. And
he's probably, just real quick on it, he's probably, I mean, you can correct me if I'm wrong, he's probably most known for the like enantioselective epoxidations, right?
Absolutely.
Probably what he's most known for.
Yeah, I mean, Jacobsen epoxidation, known reaction. Right. also his HKR react, the hydrolytic kinetic resolution. I mean, that was really big. Essentially all using, earlier, using metal saline catalysts. I mean, a specific manganese or chromium atom, right? But his approach has always been very deep thinking, I would say. He's always been focused on the mechanism of reaction, right? I mean, how do the individual molecules and the atoms interact in, the sequence of events that leads to changing one molecule to another, right?
Real quick on that, every professor I've talked to at the very high end always have a deep understanding of mechanism. That's a common theme that I've seen over and over again.
Yes.
And oftentimes,
think about it.
Yeah, you have to.
You have to. Yeah. I mean, and in total synthesis, what the reaction you're running, what is the mechanism, right? How does that affect? How can you, influence it based on the conditions of the reaction, right? One of his big things also has been, an anti-selective catalysis. Okay. Right. And, it's this idea of chirality, right? Molecules can be mirror images of themselves, like our hands. I mean, this is what everybody always says. And, being able to, in a reaction, make just one of those versus the mixture, right, is really important. I mean, right. These molecules of nature, these natural products that people work on, I mean, most of the time they exist as a single hand, right? Bodies are made up of these biomolecules that all are, a single mirror image isomer. So being able to selectively make one of those is really important. It's been a very important pursuit. And so that was something that, Jim Leighton was interested in, and I thought about that some there. And Eric Jacobsen has been, he's one of the giants and sort of pioneers in the field of asymmetric or anti-selective catalysis and studying mechanism of that. And he also, so the science was just appealing to me. I want to do something a little bit different than what I did in my PhD, right? I wanted to learn something different, which I think is really important. That's part of the reason why people do postdocs. But he also, it was clear to me that, I came from, I was with Phil, and he was a fantastic advisor. And so, I looked to Eric, and he has trained a lot of professors, people that have gone on to be professors. And that said to me, well, this guy is a great mentor, is a great trainer for people that want to do that. And so, just like my PhD, where I said, here I'm going to learn the most in this opportunity and, help me be the best chemist I can be. I saw that as a good sort of jumping off point into my independent career. And I was very, I'm grateful for the, all the experiences that I've been able to have. I feel very lucky. And so I was lucky enough to be able to work with Eric and develop a catalytic reaction, asymmetric reaction. And he gave me a lot of independence, which was great. I think as a postdoc, you want to be independent. Of course, I couldn't have accomplished what I did without him by any means. And it was based on chemistry that he had developed, right? But, I think he wanted to see, can this guy, get something to work on his own in this period of time? But yeah, he really advised me on how to go about it. Just like with Phil, how do you develop a chemical reaction? I didn't really know how to do that, and really think deeply about mechanism. So I really learned a lot about how to think about mechanisms, how to sort of evaluate them in Eric's lab. And my independent career is really, I think you can see it. really, influenced by my PhD and my postdoc. That's, some people,
the natural order of things,
right. I mean, it really, it is, I mean, there are some people that, more their postdoc, they're in that area or just their PhD, but I was really I really consider myself a combination of the two. I mean, really,
I mean, this brings us nicely kind of into your independent career. And ultimately, why I really wanted to talk to you, because I just think it's the things you do now is just really, like I said earlier, just eye candy, just really cool.
Thanks.
And so, I mean, I'm gonna kind of let you take the floor here. And there's like 3 specific publications that I'm really interested in, like those the nitrogen atom insertion.
Yep.
The making like those. I guess I think there's the right nomenclature, like those dicyclobutadienes. Your most recent, like Jack's Com, I think that's the right language.
Using cyclobutadiene to do these kinds of cycloadditions and
then making, and then those I'm forgetting. It's almost like those pyrrole. You take those aliphatic or allo nitrogens and then making like those cycloadditions. Yes. Right.
Exactly.
I think it's right nomenclature.
Yeah. We can take like dialamine.
Yeah.
And then do cycloadditions with the 2 double bonds with the alkanes, right, to make cyclobutanes. I mean, we've done a lot of cyclobutane work in my lab, and it really came from these weird lipids that we worked on early on. These so-called laterane lipids, which, I mean, you said it, are like real sort of eye candy.
Yeah.
And, I've always been drawn to, I talked about some of the purposes of total synthesis, of, trying to make a compound that, might have interesting biological activity and could, help treat human diseases. But, on a fundamental level, I mean, I've just always been drawn to interesting looking structures, right? I mean, there's a real aesthetic side to organic chemistry and the structure of molecules, right? And you see enough molecules, you get a sense of, what's common and what's not common. And a lot of times, uncommon types of structures are very interesting. I mean, and they're very inspiring for new chemistry, because it's not necessarily clear that how to string known reactions together. So it forces you to invent new chemistry, right?
Yeah, so let's start there though, because those laterane natural products that you have.
Yeah.
So like how did you come across those? I guess you and you kind of mentioned it too, I guess, is that at a fundamental level, chemists are interested in just making this thing, right? Like, let's just say money weren't an issue. Like, dang, I wonder how you can make that. And so I'm curious to know, one, how you came across those sorts of types of natural products, and do you know how in nature like those types of structures are made? Or
we still don't know that. We still, I assumed that some point in the last 10 years people would have figured it out, but people still haven't figured it out. And that's a big area of research, is just figuring out, not just making things from scratch, but actually figuring out how nature is putting things together, right? That's a very, very important area of research. We don't do that. That's more, I mean, you have to, understand the biology and enzymes and how to work with those more. That's not things that, we do. I came across them during my postdoc, reading literature, and there were some papers by E.J. Corey, who's a Nobel Prize winning organic chemist at Harvard. And he had made these things, and I was just fascinated by the structure. I just thought, that they're highly strained molecules, and strain is energy, and so that means that an organism is expending a lot of energy to make these things, and that says there's some important purpose to them.
Right.
I mean, I like to say, nature doesn't do things just for fun. I mean, nature makes things for a reason. But so, I mean, I was originally drawn to the structure. As I said before, it's kind of hard to justify just making things for the sake of making things. So what I did was, and I give this advice to everybody, is, I really dove into the background of these structures. And what became clear to me is that people didn't understand why these things are made or what the properties of these things are, right? I mean, nature makes molecules and they have certain properties. And so what was this? I mean, they're lipids in that they make up a, cellular bilayer, and they don't look like any other lipids that we've ever discovered. And so because of that,
just how so in their chemical structure, like the chemical structure of the laterin is just unlike any other, let's say, lipid.
Exactly.
Chemical structure. Okay. I see.
Exactly. A lipid or a lipid that's in a bilayer that makes up a membrane, the way we sort of, they're introduced in early, biology classes or chemistry classes is that, you have this long sort of greasy, hydrophobic, we say, water hating tail. And then you have a, hydrophilic or water loving head group that's attached to it. So you have, something that likes water, a long thing that doesn't like water. And so these things self-assemble where all the, non-water portions come together. And then you have these water loving things out on the sides. And so you can have aqueous environments on either side, but then you've created this barrier that doesn't like water. So it can prevent things from going in and out. Right. And that is fundamental to life.
Yes.
These questions of, I've always loved the, origin of life chemistry area and reading about that. And, at some point, chemical reactions are happening, but to make an organism at some point, you need to compartmentalize them. Yeah. So,
and that's real quick on that. It's interesting though, because, many lipids that have like the hydrophobic chain, they're just a chain, they're just aliphatic chains, they're just a single, they're a single chain. They're not in this ladder like structure. So that is, now that I'm thinking about it, I'm seeing why you got interested in this, because why would nature need to make that? It seems like it would be a higher energy cost to make. So exactly. It's that's actually really cool.
Exactly. Exactly. Right. Normal lipids are just these one atom, one carbon atom attached to another, and they're just these long linear chains. But the laterins have these folded up structures that's composed of 4-membered rings all together. That's why people call them ladders, because if you draw it, it's just squares, together. And because of how carbon bonds to things, if you put it in a 4-membered ring, it's not very happy. again, there's this sort of strain. They're stable. They don't just explode, but they're, they are stable. But as we've been saying, making those, nature putting that together, introducing that strain, that takes a lot of energy. And the organisms that make these don't just make a little bit of them. They make up a huge percentage of all the lipids that they have. So again, tons of energy is going into this. And that allowed me to say, well, there is a reason to make these, and it is to actually study their physical properties, which is different than your sort of classic total synthesis, which is, as I described it before, making a molecule that only arises in small amounts from an organism and that can treat some disease, right? That can kill cancer or kill bacteria or something like that. And so this, sort of seemed to me like a new idea, new way to justify making molecules of nature, which is to study their physical properties versus their, biological properties, if you will.
So is that what you're kind of doing now with these, I guess, part with these laterins? Are you doing the understanding the physical properties of that, or is it?
No. So this is where, collaboration really comes in, because doing the physical chemistry experiments, really precise measurements on these things, that is not something that my lab has expertise in. And so we needed to collaborate with somebody. And, one of the reasons that I came to Stanford is because I got really excited talking to Steve Boxer, who's a professor here, and he's a physical chemist. He's not an organic chemist, but they've studied membranes a lot and done physical studies on them. And so I think, maybe one of the reasons I got this job is because I was able to get, a physical chemist excited about.
That's beautiful, man. Sometimes you got to play the game a little bit.
Yeah, no,
Yeah.
totally. It's a
man.
part of this, for better or worse.
Yeah. That's really cool, though.
But collaboration, everyone talks about collaboration, and collaboration is very important for science.
Now, hang on. So with these laterans, though, and is it? I guess this might be a little bit too generalized, but the laterans that you're you were looking at, is it? You may not understand the physical properties yet, but is it are the biological mechanisms of these, like, let's say, lateran lipids? Is that understood? And where do these come from? Like, what sort of organisms are these?
So they come from a bacteria. They're so-called Anammox bacteria. That stands for anaerobic ammonium oxidizing bacteria. Okay,
makes sense to me, but
yeah, they grow in environments with very little oxygen.
Okay.
And
what's considered very little oxygen, though? Like, what is considered? Like, what's considered very little oxygen?
Geez, I mean, as far as a sort of ppm number is concerned.
No, just like not atmospheric, I guess. Like, not like
right. Sorry.
Okay.
So these things are found in water, in aqueous environments.
Oh, okay, that makes sense. Okay.
And there's lots of aerobic bacteria and organisms, and so they tend to live in these environments where there's very little oxygen, and it could be because other organisms are just sucking up all that oxygen. Okay. They were originally discovered in wastewater treatment facilities, actually. So poop. They live off of nitrogen.
You can learn a lot from feces.
A lot of nitrogen.
You can learn a lot from feces, man. Don't be sleeping on the feces, man. Although I wouldn't want to do that job. But
no, not at all. Not at all. So there's actually a real practical use for these bacteria. I mean, they have developed in Europe, like, wastewater treatment facilities that purposefully use these things now. So yeah, I mean they spit out. So they use nitrogen sources for energy, and they spit out dinitrogen, N2, which makes up 70% of our atmosphere. And the whole nitrogen cycle is very important. I mean, taking N2 as an energy source and fixing it, right? I mean, the whole nitrogen cycle is one of the very important, chemical cycles on Earth, right? For all the living things. So they produce a lot of nitrogen, but people took these bacteria and extracted things from them, like one does to look for natural products, and they discovered these really weird lipids and solved the structure, showed that they existed, and I think people were really, really shocked. I mean, this was the Nature paper. It was a very important paper, where these chemical structures people had never seen in nature before. I mean, really bizarre, alien.
Wait, there was a Nature paper on, like, the discovery of these.
The original isolation. Yeah.
Oh, okay. The original isolation
of these molecules.
I see.
But it was these papers from Corey where they had made these molecules that really got me excited, and digging more into the background and literature, I realized that having more of these things would be very, very important.
For sure.
I mean, despite the fact that these bacteria are everywhere. I mean, they're very hard to grow because you have to grow them in the absence of oxygen. They grow very slowly, and they produce a wide diversity of these lipids. So getting out, decent amounts of, like, pure samples was an issue. So we said, okay, let's try to make these in a really efficient manner. I mean, E.J. Corey had shown a way to make these, but we wanted to come up with a, hopefully more efficient way of producing them.
Well, that brings up the natural question then. How do you make these things? Because I was looking through your Jacobsen paper, and just kind of beforehand, and like, I saw like something about Musa. I'm mispronouncing his name, or the original precedent using like an iron catalyst. But so you had that precedent, but also like what was the optimization to get those to work, and like so how do you make these things?
Yeah. I mean, our key kind of disconnection was to essentially take this ladder structure and chopping it in half. Okay. I mean, when people do total synthesis, they do this so-called retro synthetic analysis where, you take your desired product and you say, okay, what could I possibly have started with to get there? And that, that analysis is the big part of planning of how do you put together a molecule from simpler things from scratch. And so we essentially chopped it in half and then looked at the two individual pieces, which were also known compounds, not so easy to make. So we came up with a new way to make these, and this is this so-called bicyclohexene, which is essentially just two 4-membered rings together. And then if you take two equivalents of those and bring them together, you can get 5 4-membered rings all fused. And that's like the kind of key structure. Recognizing symmetry in molecules is an important part of the analysis in natural products or molecule synthesis. These molecules are clearly kind of have symmetry, certain types of symmetry. It's not always true with other molecules, and some really exciting, or really interesting, total synthesis projects have kind of found hidden symmetry in molecules. But the symmetry here was pretty obvious. But it was also very sort of enabling and simplifying because, okay, we could, find a new way to make this smaller piece, bring it together, and then all of a sudden we have this, 5-cyclobutane, pretty complex structure. And then we use some other sort of modern chemistry to functionalize one end of it, do a so-called, CH functionalization type reaction.
Have you noticed like the relative stereochemistry of these things? Like I know like they're kind of like almost zigzaggy.
Exactly.
But could you get it like maybe it's a plan for the future, I don't know, like could you get it to go like I don't know, like something like that?
Absolutely. No, absolutely. And that is a question of like how do these things come together? Because if two of them come together like this, then you can get a sort of zigzag. But instead, if they come like this, then you get like a kind of U-shaped. And, it wasn't super clear to us that we would get one or the other.
Yeah.
I think, we got a little lucky. I mean, there was reason to believe we would get the one we wanted, but I mean, that's one of the fun things about chemistry is that, you plan it, you don't know. And when it is successful and it is something that you predicted, you feel great and you feel smart. But, if it's something you didn't predict, I mean, that can be very insightful and it can lead to other discoveries and ideas, right? When things go wrong and you pay close attention to the detail of what went wrong, right? It can lead you in places you didn't think you would go. In this case, we got what we wanted. But you said it. I mean, a structure like this is going to have different properties than a structure like this.
Yeah.
Right? And actually, we have made that compounds like that, and
cool.
They end up being a lot less stable for some reason. And so, it's interesting because it's like, well, nature evolved to make this structure, and it is more stable. So
interesting.
Is that why nature did it? I don't know, but
yeah, who knows?
No, it's interesting. It's very interesting.
The next thing I want to talk to you about is your the nitrogen insertion carbon deletion reaction.
Sure.
That one's really sick. But first, I need to use the bathroom. I really need to take a piss. Hang on, folks. I'm gonna be right back.
Okay. Yeah.
Sorry about that, folks. I'll cut that out. I'm gonna cut that out. I really had to pee so badly. I've been drinking so much water. It's crazy.
It's good. It's good to drink water.
Real quick though, I was actually just listening to Andrew Huberman's podcast about hydration. So like, I mean, have you met Professor Haouamine at all? Like, just very curious. Okay, I'm sorry.
Well, no. I mean, what was it? I'm curious. What? what were they talking about? Or
within the podcast, about like for hydration or just like, or
yeah, for hydration. Yeah.
Oh man, he broke down it was kind of beyond me. It was like, I try to understand it. It helps with your skin, sleep cycle. I don't know. But I was curious to see if you ever like see him around campus at all. That'd be pretty cool.
I'll look out for him. I don't know.
Anyway, I want to get back to the nitrogen insertion, carbon deletion, because I think from, I guess, for a non-chemist to be able to just switch out, like, let's say a carbon to a nitrogen may seem simple, like on paper, but like synthetically, it's like extremely difficult.
Yeah.
And so very curious to know, like, what was kind of your inspiration to do those types of reactions?
Sure.
And like, ultimately, like how you ended up doing it. So I'd like to hear about that.
You said it, like on paper, it's kind of a simple, for a non-expert, like taking a simple type of molecule. It's a benzene ring. It's 6 carbons all together, very stable. And then turning it into a molecule that also has 6 atoms in a ring. But one of the carbons has been swapped for a nitrogen. Practically speaking, that is really, really, for people that know chemistry, that's a really big challenge. Yeah. And it's been something that, has kind of been in people's mind for some time. In the half of our program where we are doing, it's not the same as what Eric Jacobsen does, but, it is methodology development of one single reaction. We've always had projects like that going on. And so this is one of those, right? How do you specifically do this transformation from one known thing to the other? And, I actually want to say it was in one of my first years as a professor when I was, I visited Merck in New Jersey, and I gave a talk there. And somebody mentioned, they had one of these like conferences or meetings where I think they called it like disruptive chemistry, which is essentially like them trying to come up with ideas for, reactions that don't exist that could be really enabling and exciting. And they, and I don't even remember who it was, unfortunately, but they said, oh yeah, this is one of the things that came up. And, specifically the carbon one is called, benzene, and with a nitrogen, it's pyridine. So this benzene to pyridine idea. But that was a while ago. That was, 7 or 8 years ago. Mm-hmm. And, that's what you do over time. You kind of pick up ideas and areas and, they sit on your computer or something. Like, this is something that would be cool. And so, I thought about it over the years and pitched it as projects to graduate students to think about. And it wasn't until, recently that one of my grad students, his name's Sajan Patel. He's now a chemist at Gilead Pharmaceuticals here. So working on, next generation of medicines. It wasn't until he really decided to jump into it. And the kind of key thought here or idea was that, well, okay, maybe we can't go directly from the benzene to the pyridine, but if we can go through an intermediate, some sort of intermediate, okay, maybe that'll kind of give us an entryway. And so, we found, chemistry is based on precedent and what people have done before and what you can find in the literature. And
some German or Russians done it before.
Yeah, no, exactly. Phil Baran one time sent me an article that he had found in a Russian Journal of Chemistry. Like I think he just went to the UCSD library and was just looking at old Russian chemistry journals because there's a lot of stuff back there, right?
And like just as a little bit of a tangent, but like pre World War II, World War One, like Russians and Germans tried everything. Literally everything. Like if you can think of it, they've done it.
Very true. Very true.
But anyway, sorry.
safety aside, right?
Well,
do anything.
well, they washed their hands with benzene. It was all good. It was all good.
For sure. So yeah, I mean, so he saw this precedent where they had gotten trace amounts of our desired pyridine. Like they're mostly making this. So essentially, it took the 6-membered ring with carbon and generated a very reactive nitrogen atom off of it that then could insert and expand it to a 7-membered ring.
Mm-hmm. Like azepine, right? Is that what it's called?
Yeah, exactly. Azepine.
In your case, was it conjugated, or I guess not conjugated, but conjugated would be the right word, I think. Right?
There is conjugation. Yeah, there is conjugation in the ring. And again, they were trying to make the azepine, really. But they found trace amounts of this pyridine. And that was it was like, okay, well, maybe we can find a better way that kind of follows a similar idea to get from the benzene to the pyridine. Mm-hmm. And it's kind of wacky chemistry. I mean, it uses light. So using light in chemistry, right? So-called photochemistry. Yeah. We've done a lot of that through the years. I mean, it's a very hot area of research. I mean, it's always been part of synthetic chemistry, I'll say. But it just allows you to do some really crazy things. Because when you shine light at molecules, light is energy, so you can get them to these high energy states where then, crazy stuff can happen, that you couldn't do by just heating up a molecule. Mm. And so it generates that very reactive nitrogen atom with light that then can insert to make this 7-membered ring. And then, the real challenge was then how do you take the 7-membered ring and ring contract it with removing a carbon, right? You inserted the nitrogen, one step, opened the ring up or expanded it, and then you need to excise a carbon to get there. Yeah. And he really found a way to do that was different from the way that they did. I mean, our proposed, mechanism that we, again, proposed based on some studies we did is different than the mechanism that they kind of proposed or thought of. I mean, I should say, sort of backing up a little bit, I mean, big picture, there's a reason why maybe people at Merck, and I'm sure other people sort of thought, oh, this could be a good thing, because if you look at, the pharmaceutical literature, there are people, there's one paper that we really kind of quote, which talks about a so-called necessary nitrogen effect. And I don't know if it's necessary or not, but there
sounds nice.
It's if you take a ring that only has carbon and you introduce a nitrogen into it, you swap these atoms, you can drastically change the properties of that. And there's a lot of cool examples from that paper. I mean, it's like kind of a review where people have done that, and then either the biological activity is improved, or just the properties of the molecule are improved. Maybe it gets to where it needs to get better, or it doesn't get metabolized as quickly or something. But again, people hadn't been able to just take one of those rings directly and convert it into the other. So that's kind of our pitch. And, maybe a long-term goal of this project is to be able to take a complicated drug that's already been made and then swap out a carbon for a nitrogen, right? Traditionally to make the sort of nitrogen analog of a molecule, you have to go all the way back to the beginning, develop a totally new way of making it, which is a lot of effort. So that is increasing the efficiency of being able to make this one atom swap.
Real quick about that though, but how would it be easy to install the azide though? Is that something?
That's that's a good question. That's a very good question. Azide, there's a lot of an azide is a group of 3 nitrogen atoms all connected. Azides themselves have a lot of energy. I mean, sodium azide is used in airbags. It's explosive. It releases a lot of energy. So, there's some safety concerns with using azide, I will say. You can take a single nitrogen connected, not in the ring, but connected on the ring to a benzene. And you can take that single nitrogen, you can convert it into an azide.
Okay. Mm-hmm.
You already need to have the nitrogen attached to the ring in that case. I mean, there are some ways to just directly add azide to certain types of 6-membered, all carbon rings. But it takes some steps. It does. And I would say, that's definitely one of the limitations of the chemistry at this point. I mean, this is a
for sure,
step,
but yeah.
Right. And
I mean, this is cool. I mean, like, I think this is fascinating. I mean, again, we talk about it's like fundamentally taking benzene and going to pyridine is just fundamentally really cool.
Yeah.
That doesn't work well for funding, but like, I'm just, from a chemistry point of view, it's like, that's sick.
I mean, I'm working on a grant right now on it. So it's fresh in my mind. It's like, how do you really convince people that this is an important and sort of achievable goal?
Well, I mean, the framing of like the necessary nitrogen, like if you are deep into a total synthesis or like you're making your pharmaceuticals, I mean, if you could just install that azide and then switch to a pyridine within 2-3 steps, like that is instead of redoing an entire.
Yep.
Because I mean, like you mentioned, I mean, if you just start with pyridine though, I mean, that is a whole different route then than starting like from a benzenoid for sure.
Yeah. Absolutely. No, absolutely.
That's really cool. So I mean, what's kind of like your envision for the future of this? Is it like trying to like what's kind of the future of this project, you think?
Yeah. I mean, one thing is getting away from the azide. I mean, the azide is not terrible. I don't want to paint it in this light. Like it, whatever. I mean, people use azides all the time. They're great. But it'd be nice to use something, maybe do this with something other than the azide.
Sure.
Being able to directly do, use an aniline, which is, the single nitrogen atom connected to the 6-membered ring.
Yeah.
We could just use that directly. That'd be great. If we could just use an external nitrogen, so really take, all carbon, no nitrogen on it ring, and use some external nitrogen source to come in and insert. That is a big goal. I have a former student at Genentech who has been, has showed some interest here. So, I'd love to potentially collaborate with them on using different external nitrogen sources. To make,
I'm really excited for that project, and that's really cool.
Yeah. But we also just want to improve the efficiency of it,
for sure.
Right. That's always
for sure.
Very well. That's always a goal.
Yeah. I mean, when a paper comes out, it's easy to just pick holes at it, but like very easy. But it's very easy. But I like it a lot. I think it's super cool.
Thanks, Ed. It was exciting. I mean, and it's also pretty different than things we've done in the past.
For sure.
Which is cool. Which is exciting for us. Yeah.
Yeah. I mean, it's different than what anyone's done before. I mean, it's like, it's really cool. The last thing I wanted to talk to you about though is the I'm drawing a blank right now on the publication. Those using a copper catalyst to make those
yes,
I don't know, amino penta buta dicyclo. You might know what's the nomenclature here.
Well, I'll even just they're amino bicycloheptanes.
Okay. Yeah. Okay. That's much more elegant than I.
And that just means hept means 7, so it means it's there's 7 whatever there's 6 carbons and 1 nitrogen.
Yeah.
Excluding the hydrogens that are off of it.
Yeah.
But
I guess I was just going to ask like about the origins of that one, that project, and just and then I guess the optimization, like how you ended up doing it.
Sure. It definitely has origins in the early Lateran stuff that we were talking about. I mean, I talked about how there's 4-membered rings in those, and so that got us excited got me interested in making compounds with 4-membered rings. I mean, again, in just the sort of general sense, like 4-membered rings are kind of crazy. I mean, they're strained compounds and, not always that easy to make. Now, in that synthesis, we used this copper catalyzed reaction. And so it's a, photochemical copper reaction, where essentially, a copper atom comes in, it binds to the two starting materials that both have carbon-carbon double bonds. Okay. And then it turns those two, carbon-carbon double bond species into the four-membered ring. I mean, we call it a 2+2 reaction, right? And it's kind of a unique photochemical 2+2 in that the starting materials can be very simple. They don't need to have, appendages that kind of activate those two double bonds. So yeah, I mean, it's very unique in that sense. So it's a cool reaction. I mean, we didn't invent it, of course, like it's been known for a while. But the key, and so we were looking for opportunities to kind of use this and develop it further. And one of my students, who's also now at Gilead, named Carl Manson, former students, he noticed that these reactions could not be done in the presence of nitrogen. So you can see nitrogen's been a little bit of a theme in our research recently.
It's a great element.
Part of that is because, nitrogen shows up a lot in pharmaceuticals.
It's also a pain in the ass to work with,
Yeah, exactly. And so this reaction it's a very powerful reaction, but it was not tolerant of nitrogen.
Yeah.
And so he said, well, maybe we can come up with conditions that allow this to be tolerant of nitrogen. And his sort of key insight or idea was to run the reaction in water with some acid, which essentially puts an extra hydrogen atom on the nitrogen. And, nitrogen compounds are basic. And so you react them with an acid and you get a salt, essentially. And that salt is sort of protected and doesn't do anything funky with the copper. And so, it just kind of gets rid of that problem and then allows us to do it. But the really cool thing about this is how practical it is.
Yeah.
I mean, it uses copper sulfate and
probably one of the most common copper salts.
It's the most common copper salt, and sulfuric acid, which is super common, or we can use other acids. You just run it in water. I mean, the most complicated thing is shining light at it. You do need special lamps to do that. They're not super uncommon or whatever, but
What's the wavelength you guys have to use?
Yeah, we have to use 254 nanometer.
Okay.
light.
Is that I'm not really a photochemist guy. So is that was that I guess that would be ultraviolet?
Yeah.
Right.
No,
yeah. Or towards the ultraviolet end of the spectrum anyway.
Yeah. Well, it's lower wavelength, right? Then, I mean, a lot of photochemistry goes at maybe 300 to 350 nanometers.
Yeah.
So, a higher number in wavelength means lower energy. So a smaller number means, it's inversely proportional to the energy. So 254 nanometer light, it's a little higher in energy. But 254 nanometer lamps are used to disinfect water. They use UV light to kill bacteria and stuff. So
it's used 254 is used on UV for to scan your TLC plates, right? I mean, right? 254?
They have low and high. Some of them are 300, 350 also, but yeah, that may be more common. But whatever. I mean, it's not like some crazy way. I mean, it's higher in energy, but it's not like some crazy.
Right. Yeah.
We order these light bulbs. I mean, you can order tons of these light bulbs.
Yeah.
But the practicality is just really cool. That's really exciting. And it makes structures, again, it makes structures that have shown up in pharmaceuticals.
Right.
And, have not been super easy to make. I mean, this sort of parent compound, you said it earlier, this dialamine, which is a nitrogen, with two of the same sort of 3 carbon pieces off of it.
Super simple building block.
Very, very simple. And it's super cheap. And so we can make this compound. We run our reaction. We make this azabicycloheptane.
Right.
And you can buy it from specialty chemical sources, but it's super expensive. And so this is really enabling. It makes it very easy to make this compound.
Especially if your solvent's water, that's every pharmaceutical industry's dream right there. That's the only way really, if it's in water, that's pretty much the only way they're going to scale that up.
I mean, people joke, right? Like the ideal reaction is one you can just run in a bathtub with water, open to the air, the reaction occurs, the desired compound, crystallizes and becomes a solid. And you can just open the drain and drain off everything else, right?
Yeah.
That's that. We can't crystallize ours directly, but we can run in water. I mean,
I got to be honest with you. A lot of the like older heads in like total synthesis, like the real synthesis guys, they might roll their eyes at the, like, I think when Professor Phil Baran puts like the funky, like, oh, you can do this in a bathtub type of reactions. You know what I mean? And I think that's super cool. And it's like, if you want to get people excited about chemistry, like those are the types of things you got to do. Like, it's like really making these things actually applicable. Yeah. So I like those things. So
thanks, Aidan. Me too. Yeah. Me too.
Just to conclude here, really enjoyed your time on here.
It's really fun. Yeah.
A plug for people coming, looking for chemistry at Stanford, what exciting project you got on the horizon for a prospective graduate student?
I mean, the projects the Ph.D. project is there's a lot of exciting stuff to work on there. So that's like a big sort of forward-looking thing that I would love for people to come in and be a part of the team that works on that. Yeah. Along this theme of inserting nitrogen, there's cases where doing the opposite of deleting a nitrogen can be exciting and might be useful. And there's been some cool it's been kind of a hot area in the field, I would say right now. We have some sort of reactions in the works in early stages that show new ways of being able to do that. It's sort of conversion of a common, grouping of atoms called a functional group, right? One functional group to another, very common ones, but doing it in a wholly new way. Yeah. And that's the sort of methodology side of things on the, the reaction development. On the complex molecule side of things, I mean, we have some really cool natural products. They're called discorabdins, that are anti-cancer compounds. Yeah. And we've been working on them for some time, and I have a couple students that are actively pursuing that, but it's a huge family of molecules and we want to make a lot of them. Yeah. And that's a place where, we are starting and will be collaborating with, biologists to really try to understand the biological activity. I mean, these things have some really interesting potential anti-cancer activity. And so for people that are maybe interested in, not only making complex molecules, but investigating how they interact with biological systems, kind of probing that, is really sort of forward looking to, I think, what we'll be working on the next few years. But we're always looking for new projects and new ideas. I mean, we love, taking problems that maybe seem simple on paper or are, kind of known problems like the benzene and pyridine. I didn't come up with that idea. That problem was out there. And, I like taking problems that have been in people's mind for a while and then coming up with a solution for them. I think that's, I think there's a lot said in that, kind of trusting the community that this is a really important problem. Yeah. And then let's try to solve it. Yeah.
Well, it's all very exciting. Professor Noah Burns on the, actually, I don't even know what episode we're on, but I want to thank you for your time.
For sure.
It was really fun today. And, hopefully we'll bring you back on in the future. If we got some more projects coming on, we'll talk about those as well.
No, thanks for this opportunity. Yeah. I really, really appreciate it. And keep this up. This is such a fantastic thing to do, to tell the world about, what's going on in these labs. And
yeah. I mean, at the end of the day, it's like, I mean, I'm trying to show people how we can appreciate chemistry a little bit. Because I mean, like we were talking about a little bit beforehand, like it's hard to kind of market chemistry, I think, a little bit, especially like when you're talking like, I don't know. I think it's easier to like market like a biology thing or like even like physics, like astrophysics and stuff like that.
Yeah,
so,
no, for sure. Well, and people think, chemistry is chemicals, and it's like chemicals have this terrible name, like, oh, chemicals are bad. I mean, everything on this, everything is made up of chemicals, right? I mean, chemicals are not inherently bad. There's a few bad actors, but that's right. Chemistry is not bad. It solves all the world's problems.
Right. And so just trying to shift that paradigm one day at a time. That's all we can really do, you know?
Absolutely, man. Absolutely.
But
cool.
Everyone, thank you for listening, and I'll see you on the next episode.
All right. Thanks, Aidan.